Yeah, so I worked on wasps for many years and was interested in questions of sociality and thought it was pretty important to discover how social they are and how what governs that. But it then turns out that really all of life is social at some level or another, and even cooperative among other species. So that's sort of the angle I'll take today. This is my group as of last year, back in the pre-days when we were all just hazardously sitting around. I can tell you that, can I use my mouse? This is Alexis, and her roommate also is in our group. And yeah, she's at home right now. Oh, maybe it's, yeah, there's Mariah. Yeah, she's sick right now. We really hope she doesn't test positive. She's sick too, tested negative. So our lab meetings these days just kind of look like this, as yours probably do too. So when you think about interactions, you can sort of ask the question of who benefits. And it's a bit of a shift in your mind to realize that really sociality and interactions are not the default. The default is really solitary living. So if you see animals in groups, someone is benefiting. Maybe they're all benefiting. Maybe they're not all benefiting equally, though. Sometimes they're interacting as these dung flies are, not because they want to benefit, but because there's a resource that they both want. In this case, it's a fight for dung that they can use for the females to lay their eggs. Sometimes benefits and costs are within families, in which case benefits don't have to accrue directly to any one individual if they're related. But in mutualisms and symbioses, often the benefits to different partners are very different. So this is a ranunculus. This is, it hosts a fly, and the fly both pollinates and lays its larvae in this little alpine flower, which be a great study system because it's in the Alps and it's lot smaller than the yucca moth system. Other interactions, such as this one with the magpie and the garter snake, you're not really going to have to spend too much time wondering who's benefiting here and whether or not it's a mutualism. Clearly, the magpie is benefiting and the snake is not. So when we think about microbes, I think it's really good to keep those interactions that one can see in mind, because in microbes, you can't really easily see what's going on. So you have to be really careful not to assume there's cooperation when in fact there's conflict, or vice versa. There could be cooperation that looks like conflict. So there's a lot of advantages to studying microbes here. These are spores of Dictyostelium, and these colored cells are Paraburkholderia, and the green ones are the food bacterium. Another nice thing about microbes is you can really get down to a very specific mechanistic level to understand them, something that's really tough to do with vertebrates or even wasps. Another downside to microbes is, sad as it is, this is not actually their natural habitat. This is the lab habitat, and there are changes that imparts, and we have to be aware of them. So we can say that eukaryote bacterium interactions are hard to understand. They're ancient, they're complex. There's multiple players. Cause and effect can be hard to separate, and it's hard to see what's going on. Margaret McFall and a whole bunch of people, maybe some of you, wrote a really influential paper basically pointing out that development starts of animals in a world in which there were already microbes. So it's hard to separate microbes from the environment and really see anything normal about any other kind of organism. To get at these sorts of things, I really like to have as simple a system as possible. I'd like a simple eukaryote, and this is the one that we use. This is Dictyostelium discoideum. And I like to study it in something approaching its natural context. For us, that actually often means we throw soil on Petri plates, so that's maybe not all that natural, but anyway. So Dictyostelium, which is what I'll be talking about, is a soil amoeba. It lives everywhere. Well, soil amoebae of Dictyostelium live everywhere. D. discoideum is actually limited to the eastern US and Japan and that coast of Asia, although it also occurs in India. So it's not everywhere, but its relatives are. There's at least 200 species, probably more like 1,000, I'd guess, if they were looked at carefully. It is a eukaryote, has a nucleus, has mitochondria. For most of its life, it's haploid, but it does have a normal sexual phase and meiosis. It has 3 sexes, any 2 of which can combine in meiosis. They're predators on bacteria. And they, most of the time, they have this amoeba stage where they're simply split by binary fission when they're large enough. But they then also have a very interesting social stage. That's really why I study it. This social stage is non-feeding, motile, tens to hundreds of thousands of formerly independent amoebae aggregate into a multicellular body that then crawls around and moves towards heat and light and ultimately forms a fruiting body. And I'll talk more about that in a bit. So they're here on the tree of life, sister group to animals plus fungi. So here they are. The red blobs are Dictyostelium. They're crawling through the forest soil, eating bacteria. And for those of you who prefer vertebrates, I just have this little mind game for you. This is a Dictyostelium, and this is a bacterium. So if you just imagine this is a cheetah, and this is a gazelle, and this is predation. See, it's gripping, totally gripping, just like that cougar that was following that guy on the trail, right, that you all saw. So when the amoebae aggregate, they run out of food, and then they actually follow a cyclic AMP trail. This is a pipetman tip releasing cyclic AMP, and they go towards it, and they also chain to go together. Move towards heat and light as this multicellular body, and then form a fruiting body. So here that shows the fruiting body formation. And here are the life cycles. This is that social one, the vegetative eating. I'm not going to talk about the sexual one. So in that fruiting body, the cells that are in the stalk have actually died to make a stalk. It's an altruistic sort of thing that helps them, helps the other ones flow to the top and then puts them in a better place for dispersal. So the world has a ton, maybe 100 labs of really great Dictyostelium researchers. They're mostly cell biologists. They mostly all work on a single strain, a single clone, descendants of a single clone of Dictyostelium discoideum collected off the Blue Ridge Highway. So, us being field biologists, we actually, one of our first things we did was we collected lots of wild clones. We really wanted to ask questions that were more natural, but we really benefited from, and still do, all the work that the cell biologists have done. So here's my husband's hand with a couple of straws collecting. I should mention that I do all my research in a partnership with my husband, David Queller. And here he is stabbing the soil with Subway straws. And then we ultimately got fruiting buzz from them, and then expected to see a whole lot of interactions among what we collected in the soil. So if we were in person, I would have you talk to your neighbors about what kinds of things you might want to know about Dictyostelium bacteria interactions. But so instead, I'll just pause for a few seconds to let you think about that. See if I disappoint you or answer the questions you'd already thought of. Well, I'm going to start simple. We did all these collections. The first symbiotic interactions that we wondered about were for the obligate endosymbionts. And there's some that are well known of protists. And so we use basic bacterial primers, and then also one specific to these, to simply see if all those wild-caught clones might have something in them that was an obligate symbiont. This really had never been looked for before. So here are some pictures of an uninfected spore. Here is a spore that's carrying amoebae-bacteria. Here is one carrying, I'm not sure where that is, neochlamydia. We only found one with Procobacter. This is Suzanne DeSalvo's work, along with Tammy Hazelcorn and some other postdocs in our group. So we sampled all the clones that we had collected, which were about 705 or so across the US. We focused on Houston, where we used to be. St. Louis has relatively few, and then places in Virginia, Mountain Lake, and some other places we collected have a lot. So here what you can see is a lot of them are uninfected with these endosymbionts. A few have both amoebae-bacteria and neochlamydia, and quite a few have more have neochlamydia than amoebae-bacteria. So Suzanne then cured them and tried to look at fitness in terms of spore production, and basically found that there was no significant effect. And yeah, granted, this is not terribly detailed of a way of looking at fitness, but it is an assay that can often show fitness differences. So I just said the fitness costs were subtle or non-existent. We couldn't really measure the fitness cost to the bacteria associating with Dicty, because at this point the association is obligate. So what we really wanted was a system where this dependence had not yet become obligate. And so we focused on another inedible endosymbiont that is not obligate and is often found in Discoideum, and it's Burkholderia. Burkholderia is a really cool genus of bacteria. It's actually recently been split into Paraburkholderia, which is sort of the plant beneficial genus, and Burkholderia, which has the nasty ones like pseudomalia and stuff that can harm mammals. So I'm afraid I'll mostly be talking about Burkholderia, but I really mean Paraburkholderia. So it's a gram-negative beta proteobacteria. They live everywhere. Some are animal-associated opportunistic pathogens. So here's the phylogeny, and here's the two splits. Anyway, we did just name the 3 species that are facultative endosymbionts in Dictyostelium: Agricolaris, Haliella, and Bonnia. So these Burkholderia have mechanisms that let them live inside of host cells. So we don't know all the ways they get in, but one way is that they get engulfed with phagocytosis. And normally, if it were a food bacterium, that phagosome would then fuse with the lysosome, would acidify it, and the contents would be digested. The Paraburkholderia has mechanisms to make that fusion not happen. It also has mechanisms that can confer virulence of various sorts. So I'm not breaking this one up by species, but of the populations that we sampled, again, this is just lumping them more than the earlier figure, half to a third of the clones we collected contained one or another form of Paraburkholderia. So they're pretty common. If you then try to break it down for Burkholderia along with the 2 endosymbionts, you can see that I guess the majority have just one of the 3. There were 9 clones that had all 3 genera. Anyway, it's doesn't seem like there was a lot of competitive interactions among them. And furthermore, other work Suzanne did show that the obligate endosymbionts didn't change the impact of Paraburkholderia on D. discoideum using spore counts as a measure. So this is just the first column in a panel is untreated, so carrying Burkholderia or these 2, all these various species, cured or uncured from the endosymbiont. So you just see these 2 in each panel are pretty much the same. So it was kind of relief because it meant we could just sort of ignore the symbionts for the part of the research where we focus on the facultative symbiont. So these 3 newly named species are quite highly diverged from others. Agricolaris lisa, this is fungorum down here. Agricolaris also shows the most diversity within it. These 2 species, Bonnia and Haliella, they were really different from essentially this whole group. Well, this group here is their closest ancestor, and they're highly diverged also from each other. So the first thing we noticed about this whole interaction with bacteria was actually not that Burkholderia was in there. What we first noticed was that some dicty could carry food through the spore stage and others could not. That was the first thing we noticed. It actually was the second thing we noticed, that Burkholderia carriage caused the food bacteria to also be carried. So here, this is a figure, it's a little complicated, that simply shows this. Farmer clones here in the light gray are clones that had Burkholderia in them. And then the black ones are clones that did not have it in them. And so what we were asking was whether having Burkholderia made them stably able to carry food bacteria. So here, just in this first column, you can see that if we added the food bacterium Klebsiella pneumoniae to the farmers, they all carried it because they already had the Burkholderia. But the ones that didn't have Burkholderia could not carry the food bacterium through the spore stage from one generation to the other. And this was basically true for all these other bacteria, E. coli, Bacillus subtilis, Staph aureus, Serratia maricens, and Puruginosa. On the other hand, these here are all, the bacterium we gave them was both the food bacterium and then also some of these various Burkholderia. And that all turned them and so once the Burkholderia got in, they stayed there. So that was pretty interesting. And it wasn't just for one generation. We then did it for lots of multiple generations and showed that really it was just the endosymbiont Burkholderia that was able to confer this ability to carry food bacteria. Is it an evolved trait? Is it just an accident? What exactly is going on? Is it good for the dictys? Is it bad for the dictys? These are all questions that we had. So looking at these three new species, here's some things that we found. And I'll focus on well, I can talk about all of them, but B. haleyella in particular has a much smaller genome. So here's genome size. These 2 points are genome sizes. Well, here, this is genome size, this is gene count, and this is GC content. So you can see that this is haleyella, this is Bonnia. Were much reduced. By contrast, these red points here are Agricolaris, and they are not really different in genome size, gene count, or GC content from the outgroups. So then we got super interested in haleyella and Bonnia. Bonnia we've only isolated maybe 3 times, but haleyella we found all across the range. And there's a bunch of other cool stuff. So you might remember that B. haleyella or Paraburkholderia haleyella and Bonnia were very close on highly derived lines. Well, it turns out there's a huge chromosomal inversion that between the two, and there's also a lot of other differences. But so then, you know, coming from an animal behavior background, I like the organism whenever possible to simply tell me what's going on. So we wondered if the symbiotic Burkholderia were simply attracted to dicty. Now I heard some really interesting work from yesterday on that it's not really chemotaxis for the microbes towards larger things because they move so fast it wouldn't work. But anyway, that's what we looked at. We just imagined that maybe the Burkholderia would just go swimming towards the amoebae, and they would either, we know they get phagocytosed. We don't know if they also get in other ways. So my postdoc Longfei Xu, who's now back in China at Sun Yat-sen, and who last February went to Hawaii to visit his parents and for New Year, and he got trapped in a tiny apartment with his parents, his wife, and his son for 3 months. But where he is, things are back to normal. Anyway, so he had the idea that he would just do this fairly crude experiment of simply taking vials and sticking a syringe with Dictyostelium supernatant here, and then the Burkholderia here. And if they were attracted, they would swim up this needle. Well, you try to save your group from doing things that just aren't going to work. And we tried to save him from this experiment because it seemed ridiculous that they'd be swimming up needles. But of course, he did it. And it turns out it worked. And so here you can see the Burkholderia. This is Agricolaris. We changed the names around. Swam up the needle towards the Paraburkholderia haliella. That both Agricolaris and haliella swam up the needle. Towards other Burkholderias, they were a little attracted, but not much. And then towards blanks that just had supernatant from the bacteria themselves or from non-carried, they did not swim up. So we thought this is kind of cool. This to us means that the Burkholderia must get something out of the association, or else they wouldn't be going towards it. It's sort of animal behavior thinking. Could be food, could be they just go in there and eat the dicty cell out. It could be they get better dispersal in this way. And we've shown that dicty carries food and that that's an advantage, but we don't really know overall if it's good or bad to carry Burkholderia. And, the fact that those had reduced genes, first thought as well, oh, it's a symbiont, it's all good. But parasites also have reduced genomes, so it doesn't really answer the question. So looking at parasite, so these are spores, and here you can see the cell envelope is all kind of ruined. Whereas in a cell that normally carried Haliella, it's unharmed. And then we cured the one that carried it and reinfected just to be sure this wasn't, just because that's how they were collected. And also, okay. So I was talking about these envelopes, and that the Haliella destroyed and naive one was fine on a strain that had been collected in the wild, either carrying or cured and then recarrying. Then these pictures down here, on this one, these are stalk cells. So you can see the stalk has lots of Burkholderia in it, but it has its regular structure more or less, whereas the stalk cell is relatively destroyed here. Now, just one thing to remind you is this is a sexual species of dicty. These cell lines, there's lots of gene recombination. So it's a little weird to not even sure how to think about having lineages like this, but it could be that they spend most of their time going through simple binary fission, and that they can actually evolve benefits outside of sexual recombination. Let's see. So, here's just a slide showing, I'm not going to give you all the details, but it's just basically showing that Haliella results in, for the dicty, they have shorter stalks, lower stalk volume, and smaller sori than the wild type. So they really do seem to be harming them overall. Um, and this also sort of shows that this is naive, cured, and then those that already carried either Agricolaris or Haliella and then were cured and reinfected. So this one's a little lower. And then here, so that's for Agricolaris, but here for Haliella really had a huge cost. So those that carried it in the wild produced a lot more viable spores than did those that hadn't been collected carrying it in the wild. So then that was a lineage specific effect. Here's another one, and that is that supernatant from Paraburkholderia haliella benefits the original Dicty carriers and harms the non-carriers in as they proliferate. So when they're grown in it. So these are what we call farmers that we're carrying, and this is amoeba number relative to control, and it hurts these. So we think of it in terms of the Burkholderia supernatant as like a secret sauce that helps those from which it was collected and harms others. And then it can also, we've also shown that it can help the ones that it carries in competition with non-carrier clones. So it's definitely having a fairly profound effect on a lot of stages of the life cycle. So I know it's really confusing to listen to these Zoom talks. And one thing I really like about them is you can like take off your video and then you can, you know, lift weights or do your stretches or whatever else you might want to do. So maybe we need a little more reminders of what's going on. So Haliella, Paraburkholderia haliella facilitates food carriage for Dictyostelium. It impacts the clones from which it was collected less than naive clones. Its supernatant helps its host and harms its host competitors. It has a reduced genome and it swims towards Dictyostelium. Now, it is a facultative endosymbiont. We can grow it in the lab. It actually doesn't grow that well, but it will grow on Petri plates. So then we're thinking, well, what other tricks up our sleeves can we as microbiologists do to really look at them? And the next cool one seemed to be something to do with experimental evolution. We've done a lot of experimental evolution. I'll just tell you about one piece of it, and this is Tyler Larson's work. But basically, we felt like if we separated them and then let each partner proliferate on their own, it should disrupt relationships, making foes less powerful and friends less cooperative. So it would be a good way at getting at which of those was going on. So Tyler took 3 replicates using 3 different field collected strains of Paraburkholderia haliella, took them through about 500 generations. He used the Dicty clones that those haliella had come from, 3 replicates of them, 3 strains. So that, since Dicty has a longer generation time, about 100 generations, it's 30 fruiting body transfers. So he started by taking the wild clone and separating the Paraburkholderia from the Discoideum, evolved them for 3 months, and then reintroduced them to the wild type to see what had happened. And so what he found So here the y-axis is D. discoideum spore production. So what he found was that ancestral D. discoideum produces fewer spores with evolved Parabarcoldaria haliella. So here is the ancestor production, the three different clones, and then here is with the evolved ones. So this means that the wild P. haliella was nice to Dicty compared to the evolved one. So the ancestral P. haliella collected from Dicty had somehow attenuated their virulence from nature, but that in the lab, not having Burkholderia around, not having Dicty around, they got nasty again. So that was looking at what happened to Dicty with the two different clones. We can then also look at, and so on the left panel, you can see that there's really no pattern in what happened with Agricolaris, that it sort of was a neutral change. So then we can also look at what happened to the Paraburkholderia. And what we found here was that the ancestral Paraburkholderia grows faster with the evolved Dictyostelium. So here, this is the ancestral Burkholderia with the ancestral Dicty, and this is the evolved Dicty with the evolved Dicty with the ancestral Burkholderia. So the evolved Dicty was nicer to the Paraburkholderia than the original one was. So this goes right along with the other results. It means that the Dicty was also had traits that held the Paraburkholderia back, which it lost in the experimental evolution time. So Dicty was blocking it and the Paraburkholderia was attenuating. So taken together, this implies that the Paraburkholderia ancestrally infected D. discoideum to a gain to Paraburkholderia. Dicty pushed back and the Paraburkholderia attenuated. So this is pretty cool. We've sequenced all those genomes and are now looking for exactly what has changed in both the Dictyostelium and the Paraburkholderia. So we're just in the middle of that. Actually, genomics is a great kind of project to have if you're working from home. So I've talked about costs and benefits to the Dictyostelium with this symbiosis. An obvious benefit is that they can carry food bacteria through the spore stage, which they can only do when they have the symbiont, which is itself not edible. The supernatant from it hurts competitors, another benefit. And then experimental evolution indicates a cost to D. discoideum, especially with P. haliella. Well, what's a bacteria get out of it? It gets transported along with the Dicty spores, which they're sticky. They're not windborne, they're sticky. They stick to little salamanders and flies and other multicellular organisms that crawl through the forest floor. The reduced genome of Paraburkholderia haliella and Bania indicates some kind of dependence, also lets them proliferate more quickly. That they swim towards Dicty indicates that they want to be there, so it should be an advantage. And then the experimental evolution indicates that the Paraburkholderia haliella symbionts at least have attenuated some of their virulence towards Dicty. So that's my story. I wish I were hiking in Peru, but I'm not. Just here talking with you. So thank you for your attention. glad to take questions.
All right, great. Thanks, Joan. Sorry, everybody, for the technical interruption there in the middle, but so if you have questions, I guess let's say, looks like Karen's got her hand up. You can feel free to raise your hand on the participants window, or also if you throw a question in the chat, we can try to get it addressed as well. Karen, you want to go ahead first?
Sure, great, thank you. Wonderful talk. I'm wondering if you know anything about how Burkholderia might transmit to naive Dicty cells, and whether it might possibly like spread within the fruiting body, and so maybe carriage in the fruiting body would be also advantageous as a transmission mechanism for the bacterium.
They do proliferate inside the fruiting bodies. Yeah, they do increase in numbers in the fruiting bodies, and I think this for sure has a cost to the spores. It turns out that the food bacteria, I showed them inside the spores, but mostly they're actually outside the spores, and the Paraburkholderia are mostly inside the spores. So we haven't really worked all that out. We've seen Paraburkholderia be phagocytosed in, but we don't really know if that's the only mechanism. And actually, some of these details are things that Suzanne DeSalvo is also working on now. So it's wonderful to have other people help figure stuff out.
Okay, I think the next in line was actually Monica, who put a question in the chat. Joan, I don't know if you can see it, but I'll just say it for everybody. In Dicty, how is the determination of which amoeba cell becomes stalk or fruit work? Is there a competition among them to be the fruiting body instead of the stalk? How long is Dicty's generation time for the evolution experiment? I guess there's a few pieces in there.
Okay. Basically, that's my other talk. I have a symbiosis talk, and then I have a cheating among different Dicty clones. So there's a lot of competition to be spore and not stalk. I guess the big picture answer would be that most of the time they're nearly clonal or clonal, in which case there'd be no competition, but they're not always clonal, and there is competition. Weaker cells become stalk and not spore. We've seen some evidence that different genes are turned on in chimera, and that these genes are rapidly evolving. So it seems like there is evidence for arms races. There's also a very nice set of kin recognition genes, the tiger genes. And if you look early on in the aggregation, you can see them separating very nicely, but then they all fuse again. So the fruiting bodies are not so separate. So it's kind of a puzzle. Why would you have kin recognition genes if you don't use them? And our thought is that those are the ones that tell you you're in a chimera and you'd better start fighting. The time it takes to go from a bunch of amoebae that have reached a density and run out of food to fruit, well, that happens in less than a day. But if you simply plate, 1,000,000 cells on a Petri plate, it's like 3 days and you'll have fruiting bodies. Did that get all the pieces?
Yeah, so we've got a bunch of questions. We'll see if we can get through them in the time we've got left. I think maybe it was Caitlin and then Merlo mentioned in the chat. Caitlin, do you want to go first?
Yeah, sure. I was wondering if once an amoeba is a farmer, if you see it kind of cycle through farming, non-farming stages, or once it has an endosymbiont, does it maintain that strain of that endosymbiont indefinitely?
So basically, once it's got it, it's got it. They can lose them in the freezer, we have discovered. So the longer things have been stored in the freezer, the less likely they are to have their endosymbionts. On plates, naturally, we've never seen them lose them, but we can pretty easily cure them with antibiotics. It's a little puzzling because when you look at the pictures of the spores, you don't see the endosymbionts in all of them. And so, we've thought, well, if you plate them out really sparsely, maybe some of them don't have it. But we haven't really done that. It just seems like basically when a clone has it, they keep them, but they could easily lose them.
All right, Murillo, do you want to go ahead?
Yes. Thank you for the talk. It was great. I was wondering, did you guys at all study the interaction between environment and the host microbe interactions? Like why aren't they in all dicties or in, yeah. So where are you?
So we don't really know why they're not in all of them. The place where D. discoideum is the densest is in sort of the acid high elevation forests of like Virginia, Mountain Lake Biological Station and stuff. And there, about a third of them have them. You can collect from 1/5 of a gram and have some clones that have them and some clones that don't. So we haven't done experiments or we've tried to add different things to the plate and see what happens or anything like that. But anyway, I don't know if that answers your question.
Yeah, thanks.
Okay, I think it was Andy and then Killian, and then we'll probably wrap up for this session.
Thanks, that was really fun. One question that I have is when you go around and collect different clones from sort of different parts of the United States and you see that they're associated with different endosymbionts, do you know anything about the host level variation? Like, how genetically distant are these clones that you're collecting? Do they themselves have very different gene repertoires? Do we know anything about what's happening on the dicty side?
Yeah, we know a lot about what's happening on the dicty side. So there's not really, across the eastern US, there's not really any population structure. They're sort of seem very broadly scattered. Genetically, there is a little population structure. I think we have one paper on it, and I can't remember if it showed anything exciting. We have sequenced with a group in England about 500 of these clones. So there was a group in Brazil that was going to do really detailed, population genetic, population structure stuff. They haven't. I don't know if we'll do that or what, but yeah, there's not it's not overwhelming structure at all.
Okay, maybe last question here was in the chat from Killian. For wild collected dicty clones that are found to only have one bacterial endosymbiont, can they be infected by other types of endosymbionts, or some types of dicty more permissive or restrictive specific endosymbionts? Do you know what may regulate this?
So basically, if we dump a new endosymbiont of the facultative kinds, if we dump them on the dicty, they get in and they stay. And no, I have no idea how it's regulated.
Okay, well, I think that just about does it for our time. But again, if anybody has other lingering questions, feel free to stick around for the coffee chat, and everybody join me in thanking Joan for the seminar with your emojis or your hands. Cool, great. And yeah, so I think for those who are going to stick around, maybe let's take 5 or 10 minutes, go get yourself a snack or do some push-ups or something, and we'll come back to the same Zoom